The term monoclonal antibodies essentially refers to antibodies derived from a single set of cells. The way it works is we take a cell, duplicate it multiple times, and utilize these replicas to produce antibodies. And the result are monoclonal antibodies ready for extraction and utilization. Now let's just have a quick review of what antibodies are. These are compact proteins crafted by our white blood cells, specifically the B lymphocytes or B cells. Their primary function is to aid our immune system in combating diseases by attaching to foreign materials known as antigens. Picture these antigens as small molecules on the surface of bacteria or in the cell walls. When B cells identify these antigens, they go into overdrive, generating a multitude of antibodies represented as tiny Y-shaped structures. As these antibodies disperse, they can bind to bacteria carrying the specific antigen. If another bacterium of the same type possesses identical antigens, the antibodies can then bind to those ones as well. And before we continue, make sure to like and subscribe if you are finding this video useful and would like to see more of these. Now, let's delve into the production of monoclonal antibodies in a lab setting. To produce them on a large scale, we need numerous clones of B lymphocytes. Since these cells don't divide rapidly, we pair them with fast dividing tumor cells. The fusion of B cells and tumor cells results in something known as a hybridoma, which not only produces antibodies like B lymphocytes, but also multiplies swiftly like a tumor cell. Allowing these hybridoma cells to divide in a petri dish yields an array of cells producing identical antibodies, which we can then gather and purify. It's crucial to know that obtaining the right B lymphocytes that produce the desired antibodies involves injecting an animal, such as a mouse, with the target antigen. The mouse's immune system responds by producing B lymphocytes, which are specific to that antigen. We can then easily isolate these B cells and combine them with fast dividing tumor cells. The allure of monoclonal antibodies lies in how specific they are. By selecting the appropriate B lymphocyte, we can create antibodies that bind to virtually anything we desire, whether it's a specific protein or cell within our body, a harmful pathogen, or a chemical. Additionally, we can customize antibodies by attaching various substances such as drugs, fluorescent proteins, or radioactive materials. For instance, if we had a monoclonal antibody targeting an antigen on cancer cells, we could potentially locate and eradicate cancer by attaching drugs or radioactive material and injecting them into the patient. To test your knowledge on this, here is a multiple choice question for you. From which source are monoclonal antibodies primarily derived from? A. White blood cells, B. Red blood cells, C. Platelets, or D. Tumor cells? Comment below the answer for this. And to access summary notes, more questions with the mark scheme including this question, head to our video description where you can also book in a free taster lesson with us. Thanks for watching, make sure to like, comment and subscribe, and I'll see you on the next video.